I love that song. That's a Tribe Called Red, Electric Powwow Drum. And if you're ever looking for a song to accompany you in a workout or a stiff walk to school or all sorts of things, I would suggest that. I'll post the link later on. You can see it on the screen there. Good morning. Welcome to Friday. Where I am, I can see a sunny blue sky. Hooray. I hope there is a sunny blue sky where you are. Thank you for not leaving me hanging on the fist bump in the bottom. I'm going to save that. Oh, and I like that jelly has been turned into kind of a jack-o'-lantern of sorts. All right, stylus away, pens away. I'm going to clear all the drawings and thank you for those. And we're going to get started. Working the door, a couple more people in. Awesome. Smith can do two things at once. Right? Right? We all can. Okay, so what I wanted to start out with this morning was just an observation and an acknowledgement that some of you might have been aware that there were some troubling statements posted on our course link on Tuesday. And the comments, we've removed them. You may have seen them in the meantime and be concerned about the well-being of some individuals in the class. And we just wanted all of us to let you know that support is being offered to those who need it, and there's a follow-up underway. And in order to respect the privacy of everybody who's involved, we're limited in what we can talk about. At the same time, we recognize that some of you may be feeling upset, so if you need support, please don't hesitate to reach out to the services that are displayed on the screen there. Counseling Services offers a drop-in service in the afternoons, Monday to Friday, so today still, and Student Support Network is available in the evening. And there's also other online supports like the Crisis Text Line and the Good to Talk. So let me clear this, and we will remind you of a couple of things. One of them is that your Lab 2 quiz, I believe, is due by the end of the day today. Sherry can correct me. Hello, Sherry, good morning. In the text. And also, this posting on your homepage in CourseLink, which is kudos to your fast response on the might return, because the first one is already back, so well done you. Like Sherry says here, if you haven't already slept with your nasal strip, cozy up to it, sleep and send it in. It is not too late, but please try and get it into the mail, into that mailbox ASAP. And remember, the breakup of the placement that we had suggested was that if you're in the Tuesday AM section, across the bridge of your nose, if you're in the Tuesday PM session, across your cheek, and in the Wednesday PM session on your forehead. And I hadn't actually ever read that close to the hairline, so a little judgy, but we'll just imagine that if I was going to participate, that I had a hairline, and that's where I would put it. Today, we're going to continue with our fascination with things jelly. So the Cnidaria, we're unlikely to finish it. Today, we're going to spend some time talking about overall kind of apomorphies that characterize this group. This group that, remember, Casey Dunn, the principal author of the Dunn et al. Phylogeny, what he suggested was that if a space, when he visited in 2018 and spoke to this class, he suggested that if an alien species arrived on our planet by chance, or probability would suggest they would hit the water first, and then probability would suggest that the first life form that they would run into was a jelly, and he suggested his own biases that it would be a scyphozoan. And that's how our planet would then be characterized in international, in the Star Trek Wars space galaxy quest saga of the world, would be known as the planet of the Scyphozoans. Not enough of it is known by the general population about this, so we're going to share some jelly-based enthusiasm for you today so that you can go away to your Zoom or WebEx or Skyping meetings and over the weekend with your family and friends, and as you share some refreshment, share some enthusiasms about jelly. This is captured in, there's still some general terms and general concepts that are touched on and introduced in chapter 4 and 5 of Brusca, and then taxonomically we dive into this in chapter 7. So we talked a little bit about the jellies on Wednesday. We're going to talk about them today and then finish up on Monday. And on Monday, we're going to talk about a couple of additional things to jellies. We're going to talk about jellies, the species assignment, and how naming a species actually happens. And I'm going to tell you about what I imagine for our first lecture quiz on CourseLink in this distributed environment. So that's coming on Monday. Today, we are talking about, stop it, Smith, jumpy fingers. We are talking about cnidarians. And so this is the most diverse group that we've jumped into so far. We dealt with the phylum Placozoa, which used to be 1. You can now scratch that out and write 2. Hooray! And it will continue to change as we continue to describe more species. The cnidarians, 13,000+ species, they exist as parasites. They exist in freshwater environments. We might talk a little bit about that on Monday on the way out of the group, but principally in the marine environment. So this is the box from Brusca that you can see on the right-hand side, capturing kind of 9 characteristics or the apomorphies of this group. And I've highlighted kind of 3 of them that we're going to spend some time with. They're a diploblast metazoan, so they have an endoderm and an ectoderm, but not a mesoderm as we do. They tend to be radially symmetrical, so not split on one axis as we are, but split on a circular axis. We'll talk about that as well, because that can be slightly altered in different classes within this group. One of the really amazing things about the cnidarians is the changes that they have and specializations that they have in their relatively simple diploblast, radially symmetrical lifestyle. Is they've grown into different, through evolutionary time, they've grown into different strategies that both involve being sessile, sitting still, being anchored to the substrate and not moving, and being a radially symmetrical filter feeder, and then also being mobile and heading up into the water column. And there are some of these taxa, members from some taxa can do both. And they alternate generations where there is a polypoid. Now note the spelling, not polyploid, polypoid. And that's referring, the polyp is the anchored basal disc, little kind of, let's picture it as my hand over here, filtering. So if you're wondering what to picture there, picture if you've seen a hydra before or you've seen a coral polyp. That's the polyp. So this anthozoan-like life cycle with the coral, I'm kind of spoiler alert here, but it tends to cycle and we don't have the medusoid form. But the polypoid form tends to be one of asexuality. So there's budding or strobilation. We'll talk about strobilation in a second in one particular class of the scyphozoans. We'll talk about strobilation twice, probably today once. I'm going to show you a video, and then I have another video on Monday. Why would I show 2 videos of strobilation? I hope you'll see why in a second, because it's amongst the cutest production of biology, or it's amongst the cutest biology in the world, I think we'd have to say. So, polypoid, asexual. The medusoid is where there can be sexual reproduction and mobility. And here we go. So this is strobilation. So this is a polypoid that's sending out tiny little caps, tiny little medusoid forms, and alternating that generation between polypoid and medusoid. And these tiny medusoids we'll talk about more when we get to the scyphozoans, but I have 2 videos and I wanted to show them both because they're kind of amazing. Now back on the Dunn phylogeny, where the box of the Brusca et al. Kind of characteristics of the cnidaria was displayed, it talked about a characteristic of this group being a particular kind of stinging cell, the nematocyst, or the nidae, or the cnidocyte. So those of you who've run into jellies before while swimming, you know what this is. This is a secretion of a particular cell called a cnidoblast. It's involved in defense and in prey capture. And this is basically a simplified drawing from the 7th chapter, where you've got the cnidocyl, or this barb or hook, and then this coiled tube that connects to the hook. The operculum is kind of the cap or the opening. You've got a pneumocyte capsule here that I'll show you a different diagram in a second, but that's essentially an area where there are calcium ions pumped in and maintained so that the fast firing of these nematocyst cells can happen very, very quickly. Now notice that the cnidocyl sticks up and out. So this is embedded kind of in the mesoglia in the body wall, and it sticks out as kind of a barb into the rest of the. Let's try and imagine this. Smith does hand puppets. So there's where the calcium happens. Here's the different view, another view of a nematocyst that has fired. So you basically have this high osmotic pressure in the capsule. Then you've got the cnidocyl sticking out, the barb sticking out. There's some kind of mechanical stimulus, like you swimming by and brushing it. That operculum pops open, seawater rushes in. As that crosses this opercular membrane and enters the area with the high calcium ion concentration, that difference in osmotic pressure fires out the barb. And this happens incredibly quickly, all happening in like a millionth of a second. So we're not talking Usain Bolt, we're talking Usain Bolt to the power of 6. And we have this tiny little video here. The link I'll post up in the chat in a second. So this is one of the fastest biomechanical events in the world. This is slowed down. You can see the seconds ticking by at the top there, but at a frame rate of 240,000 frames per second. So 2 meters a second at 4,000 G. This is also happening with one of the things that's happening with that tube. The reason for the tube is the injection of venom into the site of arrival. Now, that's the tall video on the left-hand side. The square video on the bottom, you've seen before. Do you remember? This was with the ciliofferans. These are the firing of paramecium trichocysts. So these barbs that, again, respond to a lot of the same stimuli. And certain authors and certain scientists have hypothesized or tried to figure out whether or not the similarity between the nematocyst and the trichocyst is a homologous, so by shared ancestry, or whether it's a result of convergent evolution. Before we dive into that too much more, I have some questions for you, or some observations that especially if you've been to Australia before, that this is actually a news clip from not this month, but about a month ago in Australia, that as the different kind of species, the Irukandjandi and the blue bottles increase, there's always an increase in stinging and hospitalizations that happen in and around Australia. So what I would like to do is hear about if you have any jelly fish. Come on, Smith. Stinging events. Have you been stung by a jelly? Launch that poll. Tell me about it. If you haven't, or if you have, jump into the chat and give us some single word, not quite safe for work or safe for work observations of what happened when you were stung by the jelly. How you felt. Oh, stuck in the waiting room. Hello, thank you. Welcome. Smith checks the chat, has abandoned people at the door. Oh, and Sherry suggesting that lobster larva swimming. I don't know if I've got a good video of that. Strobilation it's at least in the top, let's say it's in the top 5 for sure of the cutest invertebrate things. So yes, stung in the Galapagos Island. That's a very incredibly, I am envious of the place you were, though not envious of the sting. In Costa Rica, and it felt like burning. That's often what I've heard. I have not been stung, we'll say yet. Yes, the man-o-wars on the beach. And stuck or stung while at a wedding. That's unfortunate. But well done you for going to the Grand Caymans and participating in the wedding. And it makes more sense than when I quickly read it and thought that it said that you had been stung by jellies in the Grand Canyon, which would be more surprising. So 10% of you, that's kind of interesting. Well done. 10% of you stung by jellies. I'm going to start to track that between years. Okay, I will jump back over. We did just vérifier le chat, and we'll come back to this observation that some authors have made about the similarity in structure and form in between different protozoan firing cells, trichocysts, and cnidarian cnidocysts. So what you're looking at here are microscope images of dinoflagellates, of a particular kind of a dinoflagellate, the polycricos, which has a trichocyst that, if I'm kind of talking now to the asynchronously attending people as I move my video down, you can see it in the line drawing there, the trichocyst that has been shot out, that it has the same kind of tube attaching the barb. And if we take another look, a closer look here, you see on the left-hand side polycricos, the dinoflagellate, and then the cnidarian cnidocyte. So the trichocyst is on the left, the cnidarian is on the right. And you see a real similarity between here is that they share an operculum, the stylet or the barb is shared, that there's a capsule wall and a tubule that hosts the tube that jumps out with the stylet. There's a lot of real similarities here. It's unresolved whether or not this is the result of homologous or convergent evolution. Now, if you picture in your mind the large phylogeny that we displayed while talking about the protozoans. So your brusque et al. Phylogeny that had all of the triangular colors for the different monophyletic groups within our understanding of polyphyletically of the protozoans. And we were contained within that, the multicellular, the metazoan animals. Now think about what the proposition is. If it's homologous, that would suggest that a shared common ancestor, this trait is derived from that, and that the dinoflagellates or the apicomplexans somewhere out here in the evoluoids had it, and then we also had it. So it suggests multiple losses, not just across other protozoan lineages, but across even within animals, because you see the sponges that we've been introduced to, they don't have it. The cyanoflagellates, the sister taxa for all of our metazoan animals, they don't have it either. Ctenophores, I can kind of spoil some of Wednesday and say they don't have it either. So it's been, if it's homologous, it's been lost multiple times. If it's not homologous, it's a result of convergent evolution. And we see this firing, sting, harpooning cell being the result of evolutionary, similar or stringent evolutionary pressures that say if you're going to be a relatively passive bag of either jelly or liquid floating and can be bumped into and consumed very easily, then it's to your advantage to develop this kind of, or those of you that have developed this kind of harpoon-like strategy will tend to reproduce more rapidly. It's occurred to me that I meant to put a slide in at the start to remind you that the office hours are also today. So I'm going to try very hard to remember not to shut the Zoom down. I'll just stop the video and the audio at the end of the lecture and go and have a drink of coffee for 5 minutes and come back and then have office hours here. So this, if you're interested in hanging around and chatting, that's happening today too. Now, ecologically, so I'm going to jump in between morphology, taxonomy, ecology, and evolution here, because that's one of the great things about this class is that we can jump in and amongst all of them. Jellyfish blooms. We talked about red tides, remember, again with dinoflagellates, and this is kind of the tag here. Dinoflagellates, jellies can also bloom and be produced in overabundance, and that tend to be triggered, those blooms, by a lot of the same things. Eutrophication, a lot of different nitrogen inputs, nitrogen and phosphorus inputs. So that provides a competitive advantage to the jellyfish because they tend to be have a resistance to very low levels of dissolved oxygen. And then if you combine that with overfishing and then alterations in river discharge, then the blooms can change over time the distribution of carbon and sink it deeper into the water column. And that changes dramatically, that reinforces and drives further changes and economic values of fisheries because it changes where and when they're sitting. So this was, one of you had reached out and shared the fact that in where you're from in Florida, that you were debating or hearing the fact that red tides were a political issue because of the times that they were tied, T-I-E-D, to discharge, agricultural discharge. And the fact that if you're going to propose a reduction in that, you're affecting somebody's livelihood is the feeling, and then as soon as you do that, that's a political football. these run into the same problems or the same features. It becomes a political issue as well. And in fact, as we talked about it in email, this is also multiple causes. So there's eutrophication, there's agricultural and river discharge, there's overfishing, all of those things. So we're involved. We're a common element in causing all of those three things. And then we suffer consequences down the line. So jellyfish blooms and dinoflagellate red tides. Discuss similarities and differences. Jumping back to the development, I mentioned they were a radially symmetrical diploblasts, and they settle. Remember with the porphyrin, the sponges, we talked about the coeloblast, that hollow ball of cells. That was the characteristic larval form. Now, if you were sorting through your microscope of seawater, a seawater sample, and you found a typically solid larva that looked kind of like this, ciliated, this is a planular larva. And this is kind of a larval type that appears in our phylogenetic history with the cnidarians, and we do see it across cnidarians and into some of the metazoans. So it's kind of a group that you might see on some presentation of the Dunn phylogeny, where a node where cnidarians branch off from the rest of the Bilateria, it might be called the planulozoa. So that would be a group comprised or characterized by having a planular larva. And so this kind of looks, you can see why. Remember, we talked about placozoans, the loneliest phylum of one. And I mentioned that they had been categorized in a number of different, they've been from amoeboids to cnidarians to platyhelminth worms, which we'll get to week after next. But you can see, I think, if you remember what those placozoans look like, why placozoans may have been categorized for a while as a cnidarian. And it's because of this similarity to the larval form. So the ectoderm in this case becomes the epidermis, and the endoderm becomes the gastrodermis. There we go. Move that around so you can see it later on. And diploblast, when I'm talking about that, we're a triplo, but a diploblast means two sprouts. So two germ layers, and no mesoderm as the remainder, as I'm saying us, as being one example of the remainder of the metazoans possess. Now, we're going to take a look at a lot of the cellular types and the cell walls, so different cellular types rather within the cnidarians. Now, we can see here, there's the nematocysts. Remember that harpoon cell? We can see it tied into the epidermis on the outside. Then we've got the basal lamina, this characteristic of metazoans, important characteristic differentiating inside from outside. Then we've got the mesoglia, which we'll talk about in a second. That's a really important, that's the jelly, essentially. And down here, we can see both longitudinal muscle filaments, and then also in that cross section, circular. So we've got muscles set at two different angles. So this is a muscled taxon. It's the first one really that we've seen. Neither sponges nor placozoans have muscles. So we see muscles here, and one of the first things that we see when we see muscles is we see evolution arranging them at right angles. And we're going to talk, when we talk about the movement of the cnidarian, as to why that has been selected for as a particular advantageous thing. So a diploblast with a radially symmetrical body plan. Now, in the alteration of these generations, medusoid and polypoid, one of the advantages of living either solitarily or colonially that cnidarians possessed is the polypoid can assume many, many diverse forms. And in doing that, it can assume many, many different kinds of ecological roles. You can have, on the left-hand side of this figure 7.8, your gonozoids and your gastrozoids. So a gastrozoid would be a feeding polypoid, and a gonozoid would be one that would be helping with store elements of sexual reproduction. Your dactyzoids, your gastrozoids. So there's a whole bunch of different forms and jobs that get assumed by the polypoid, but the medusoid tends to be much more simple forms. We see what you saw in strobilation gets bigger. And this is a colonial, what you're looking at here is a colonial hydroid cnidarian that lives commensally on shells, mollusk shells that are now inhabited by hermit crabs. So that's potentially, I'm excited. You can see the hair on my head stand up, or I wish you could. But that's a threefer, right? That's a cnidarian living on a discarded mollusk shell that's inhabited by an arthropod. Oh, if we could put in some protozoan, like other symbiote, we'd have kind of a pronged comparison or lifestyle for these cnidarian or for these invertebrates, and I could just teach about that commensal relationship to kind of touch on most of the invertebrate diversity of the world. Now, getting up and away from the substrate either is advantageous as a medusoid, clearly inhabiting the water column. We'll talk about motion in a second. But one of the cool things about the polypoids is that they can grow skeletons of different kinds. They can produce, hydroids can produce skeletons of chitin. Brain corals can produce skeletons, as you know, of calcium carbonate, and this is coral reefs. There are sea plumes that can produce spicules of an endoskeleton almost appearing spicules. And then there's hydrostatic skeletons that cnidarians follow up and use quite frequently. So that hydrostatic skeleton, remember, if you filled up the balloon with water or air and kind of squeezed it, and as you squeezed it, as you apply the pressure, you watch it change form. So in this case, this is a fluid-filled cavity that's surrounded by muscles that is doing the squeezing and changing the shape or the direction of the organism. And we'll talk a lot as we talk about particularly the many, both bilaterian and non-bilaterally symmetrical animals, about the importance of the hydrostatic skeleton. So read about it here. Remember it as simply as a fluid-filled cavity surrounded by muscles, and you can read about it in chapter 4 and 5, the one that talk about a lot of how things move, the chapter that talks about how things move. Now, they're also, in addition to the complexity of their skeleton, they, as you know, have a lot of foreign material. So both dinoflagellates, algae, and apicomplexans living in that symbiotically in that mesoglia, and in some estimates providing for the polyps of the anthozoan corals up to 90% of their nutrition. I'm going to take a second to jump over to the chat for a second. Oh, and I'm going to have to go back and read through these. There's lots of stinging stories. I see a question here, and I reposed. Thank you for re-upping it. Do cnidarians go through the polyp medusoid multiple times in one lifetime, or do they only go through the transition once? It's actually a kind of a philosophically complicated question because there are both solitary and colonial forms. So the question would be, well, what is the individual? That's the philosophical nature. So there can be polyps that are strobulating off of a scyphozoan, or little medusoids strobulating off the side of a scyphozoan, while there are all of the other stages existing on the same colonial organism. Polyps, once they bud, in cases where they can, in some of the classes where they can do both, they can both asexually and sexually reproduce. The sexual reproduction, I believe, tends to be gone through once, but asexual multiple times. But it's actually more complicated than it's complicated. Would that it were so simple. Everybody spend the weekend watching Coen Brothers films. So the musculature, we're going to start, this is getting us close to talking, we're going to talk about muscles and we're going to talk about movement. So polyps have these antagonistic sheets of smooth muscles. So the epidermal, the longitudinal, that when it fires, it causes the polyp to shorten, and the gastroderm, or circular muscle, that causes when it squeezes, so if I was a balloon hand, you can imagine if there were muscles around the hand here on this fluid-filled closed cavity, it would cause the extension of my fingers, which would be particularly Pan's Labyrinthy kind of a thing. But you can imagine in one case, the firing of those longitudinal muscles being maybe retract from the prey, whereas firing of the gastroderm, sending out the tentacles to help with the elongating the body and filter feeding for food. Now the medusa, their circular muscles, they have a ring of specialized circular muscles called the corona. And that mesoglea, you can see this great figure. This is, the left-hand figure is one from your text. The right-hand one is one from the older text, and I love it because it talks about the firing of these coronal muscles here, that as they fire, so this is the bell, and as they fire, they basically expel water out of this cavity, the umbral cavity, that jet propulses them forward. We'll talk a little bit more about that in a second when we have some video. Now the fire, so laying down antagonistic muscles of antagonistic at right angled muscles, circular and longitudinal, but then also this is the first taxon that we've seen, an organized nervous system. So it's not so centralized as ours in terms of its cephalization, which is a term that we'll come up with as we move into the bilateria. But they have motor and interneurons, and it's a nerve network just under the gastrodermis, and another just under the epidermis, and they're connected. They're simple nerve nets. They do have some concentrations or ganglia, and they innervate that coronal muscle, and so they can fire, and you're looking on the left-hand side at this polypoid, imagine a hydra, and with little ganglia throughout, and then on the right-hand side, the nerve rings here are the inner and outer nerve rings that facilitate the firing of the coronal muscle and the jet propulsion of the medusoid form. So a tiny video that you're going to see, or you have seen if you've been to Ripley's Aquarium in Toronto. This is just a video as we took our family there when we could last all travel freely. And looking at this beautiful medusa bell kind of moving through the water. And it's a close-up now of that other drawing that we talked about in terms of the strategies. So there's a relaxed coronal muscle and a bell that's open, and then the coronal muscle fires across that mesoglea, contracts, the water is expelled, and you jet forward. Now the question that I have for you that I'd like to spend some time talking about is what causes, we've talked about the coronal muscle closing the bell, expelling the water, what causes the bell to refill? And what I'd like to do is to have you, I've proposed some hypotheses here. And what I'd like you to do is to take your annotation tool in your Zoomlandia. I'm heading to the waiting room for a second. Yes. So what allows the bell to refill? Is it a relaxed coronal muscle? Does it happen kind of passively? Is it the contracted? Are there longitudinal, relaxed or contracted or fired longitudinal muscles that cause the bell to refill? Take a second and think about your options. And vote away. And I will drink some coffee. And then I will remember to screen grab your votes. Okay. Is that fair to say voting's done? Good. Well done. Now, so the answer is actually the elasticity of the jelly, of the mesoglea. And so as the coronal muscle fires, it compresses the bell, water shoots out, the jelly, the medusoid form moves forward, and then one of the advantages of having this elastic complex mucopolysaccharide mesoglea is that it then bounces back. It's like a perennially, the medusodnidarians are squeezing a stress ball. They look relaxed, but they're squeezing that stress ball and it's springing back. So they squirt out the water, the stress ball, the mesoglea, springs back. Now, the second most popular, the relaxed coronal muscle, I think that's a reasonable hypothesis, that if it's fired and then as it slowly returns, and you can imagine that it would eventually refill, and then selectively the difference between the hypothesis of relaxed coronal muscle is then the elasticity of mesoglea is that deeper into evolutionary time, if this happened faster, the lineage possessing that elastic mesoglea would be better at moving and more capable of more rapid movements and taking better advantage of a food resource. And the relaxed longitudinal muscle, for those of you that were thinking about that, that's also fine because we talked, in talking about the polypoids, we talked about the importance in terms of moving in and out of the polypoid being the gastrodermis or the circular muscle and the longitudinal muscle to retract from predators or to extend for feeding. So there you go. That's our thoughts. Let me, before I leave, remove those annotations. And we'll keep going. So there is the slide. That's what I just said. The elasticity of the mesoglea is the thing. We've been chatting. I've been chatting for a while. I'm going to jump back in. And I don't see any super new additions. So we will continue. And we're going to jump back. So we've talked about development. We talked about evolution just there. We've talked about morphology. And we're going to jump back and talk a little bit about relationships and ecology here. Just for a second, because I love this kind of thing. And so what you're looking at is a tiny baby lobster with a taste for jellyfish surfing. And I show this often connected, not this year, but often it's connected to a slide that you saw near the end of Wednesday's lecture where I called attention to the fact that DNA-based gut content analysis of marine organisms from fish and many, many different kinds of marine organisms has made us reconsider in the last kind of 10 to 20 years how frequently there are species that depend on jellies. In that case, it was DNA showing us that many, many more species than we had previously thought eat jellies. Historically, it was like the sea turtles eating the jellies. We know it's much more diverse than that. Now, that's a way that DNA has helped us understand that because DNA analysis of gut contests help us get to understanding who's eaten whom when the whom in case has no hard parts to survive. Now, in this case, this is one case where in the Journal of Experimental Marine Biology and Ecology, where remotely operated vehicles and technological innovations that have put us in place in situ in the water column from deep to abyssal to planktonic or floating close to the surface or near shore environments have allowed us to see how to really see, and in this case I mean literally to see and also to understand who's interacting with whom, because we're not just dealing with a survey of a taxa of a community by dredging up a sludge or a net through a water column to count the bits of things that we see. Here we're actually watching them in the same way that if we had set up a duck blind in a marsh and we could look at terrestrial organisms. So these particular lobsters are immune to the toxins, and they spend much of the time, what does the jelly get out of it? They get grooming, and they remove some mucus. And so the jelly gets groomed, and the lobster gets moved around, one would imagine. So there's some ecology and some background if you're looking to jump into more jelly land. So we've got 10 more minutes. What I'm going to talk about for the next 10 minutes or so is some specific examples of some of these major groups. Now remember this, so this is a figure from your text, and you're looking at it, and potentially in Zoomlandia you're looking at it and you're doing a face palm and you're like, oh, there's too much stuff displayed on there. Let's break it down and say we've got on the left-hand side, most basically on this clade, we have the anthozoans, so the corals, and we've got two lineages, Octocoralia and Hexacoralia. We're going to talk about those. And then we've got other lineages where uncertainty, like the myxozoans, which is a parasitic lineage of that we thought until very recently, like within 10 years, 6 years, we thought was a parasitic protozoan. It's in fact a cnidarian. The dashed line represents we don't actually know where it fits inside of this phylogeny, but we know it is part of this clade. And then we have the medusoid forms. So the anthozoans don't have a medusoid form. The meduozoans across here, the scyphozoans, the cubozoans, the satyrozoans, the hydrozoans, they do. And so what we've got and what Brusca and friends have displayed here in the numbers across these branches, you're familiar with tree-based thinking. These are particular apomorphies that characterize that group. And so what I've shown underneath in the bottom here is that the anthozoans are a lineage of cnidarian characterized by hexaradial and octaradial symmetry. So not just radial symmetry, but radial symmetry that can be divided predictably by the number of septa that we'll talk about in a second. They have a siphonoglyph, they have mesenterial filaments, and a tripartite series of flaps on the nidae. We'll break that down in a second. And remember, one of the goals that we have in this class is to approach your training and your thinking as a biologist from the perspective of many of you will probably end up working on birds. And that's a big regret to me. However, it's one where I think this is one of the chances, one of the only times in the world where some people like Sherry and I get to share with you our enthusiasms for the diverse strategies. And so there's necessarily a survey aspect to this course where we talk about all of the things. And as you begin studying for different exams or tests or quizzes, I can understand certainly that will appear stressed when you're like, I have to, there's too much to remember. And so I offer again, in the middle of this observation about anthozoan, the strategy to be that I don't ever expect you to remember all of the things. I don't remember all of the things. Nobody can remember all of the things. However, and as you're attempting to study, what you can do is think about where we have spent the most time together. And if I've emphasized something a lot in how much time as I prepare these talks with you, probably that's where my thinking is going. That's what I'm excited by. That's where I think the world is excited by. And so that's probably where some quiz questions will go. And then in terms of how the quiz questions will go, if they're going to have a phylogenetic aspect, like this, that initially perhaps you thought was complicated phylogeny, and now you agree with me that it's not actually so complicated, I'll show you the phylogeny. That will be the question. It'll say, here is the phylogeny with these labels. Tell me about this grouping. Is this where this trait, the distribution of this trait across the phylogeny, or this particular taxonomic group across the phylogeny? And then I'll ask you true-false questions about it. And then that's a segue to Monday's lecture, where I'll give you some more details about the lecture quiz. Back to the anthozoans for the last couple of minutes. There are about 6,000 species, polyps only, circular mitochondrial DNA. They have a pharynx, a siphonoglyph, cnidocytes. The septa, so this is a division of the coelentron into these vertical partitions and filaments. So this is a way that we've used, so it's an increasing in surface area. Essentially, it's an adaptation to increase the surface area. Remember, we talked about microvilli, and we talked about surface area in terms of necessarily increasing the places for the rates of reaction so that the volume, which is increasing by a cube, is not limited by the surface area alone, which is only increasing by a square of whatever the measure is. So these septal filaments, which are an outfold of the gastroderm and the mesoglia, they can number, we count them as if we're anthozoan taxonomists or systematists, and they can change in number from 6 to the highest that I have seen is 192 inside of a single polyp. And there you can see, this is a slide looking at the mesoglia in kind of purple and the gastroderm in red, and you can see these extensions, these outfolds. So the anthozoans. Now the sclerotina corals, we talked a little bit about, we talked about them actually right away in the very first lecture, because I made mention of the fact that of diversity, the diversity in a coral reef is second to none in the world. Sometimes they're compared to tropical forests, which I'm a location that I'm more familiar with. But when you think about phyletic diversity, so deep phylogenetic lineages, coral diversity can contain, of the 34 animal phyla, 32 of them can live in coral reefs, where a comparative phyletic diversity of a tropical rainforest, where I work in Costa Rica, would be 9. So tropical forests may have a large number of species, but those species tend to be predominantly composed of arthropods. And there can be 9 million species living on coral reefs. They, as we talked about in the first lecture that we shared, that they account for trillions and trillions of dollars annually. Coral reefs, the economic value of coral reefs, I calculated to be 4 times the value of Canada. So if we shut down Canada, we all went on a strike and there was no economic output of Canada for a year, that would be just a tiny, tiny fraction, of a quarter of what the effect would be if we shut down coral reefs, and affecting 17% of the protein that we eat globally. Now, shutting down coral reefs is not just a theoretical thing. We've talked about bleaching in the past. We'll talk about it again in a couple of seconds. So the sclerotina, these calcium carbonate corals, so we see that we have polyps, so living colonial, partially retracted or an extended polyp that is sitting and connected by these coelenterons. So they're not living individually, that the polyps are connected, so they are a true colonial animal. Now they're also living commensally, as we talked about when we talked about the dinoflagellates, so the Symbiodinium, living inside of that elastic mesoglia, these tiny little pockets of essentially life for the coral, life-giving dinoflagellate. We talked about that symbiosis where the inorganic metabolites from the cnidarian are exchanged and consumed by the dinoflagellate, that then is helping, because it's a photoautotroph or a photoheterotroph, it's producing goodness for the coral through photosynthesis. They live incredibly densely, these dinoflagellates, 30,000 per square or per kind of millimeter cubed. And they permit corals to live and to create their own environments, macroenvironments, and nutrient-poor waters. The dinoflagellates themselves can be transmitted vertically, i.e. from the parent, or horizontally, like in the aqueous or the marine environment. And historically, that Symbiodinium, or that dinoflagellate, we thought it was one species, and we now know that there's many species, and in fact, single coral species can harbor multiple taxa. Now, in talking about alterations, major catastrophic alterations that our species has made to the planet, bleaching, coral bleaching, where the coral polyp loses its zooanthella, loses its Symbiodinium in a period of heat stress or other kinds of chemical and mechanical stress, is called bleaching, and those events have been increasing in frequency since the 1970s. And in the Caribbean, where it's kind of farther ahead than it is in Australia, those losses, those bleaching losses, have been responsible for the loss of upwards of, in some reefs, 70% of the reef fish population. So there's no, as you've read or you've become familiar with bleaching, there's no magic temperature that causes it, but it continued prolonged exposure to increases of 1 or 2 degrees Celsius, 1 or 3 degrees Celsius above whatever the long-term annual maximum temperature is, tend to induce a bleaching event. Now, one of the things we also talked about that I'm going to bring your attention back to when we talked about the Symbiodinium was the fact that in the last couple of years, our minds have been collectively blown by this discovery out of the University of British Columbia that the tight and important symbiotic relationship between dinoflagellates and corals is actually a 3-partner tight symbiosis, and there are apicomplexans with chlorophyll biosynthesis genes that are also living inside of that mesoglia. And this is kind of amazing because it suggests, from an evolutionary perspective, it asks some questions about dinoflagellates. Remember, in the dinoflagellates, the majority of them, with the apicomplexans, the new lineage, these coral colloids, apicomplexans, the apicomplexa is nearly entirely parasitic. And this one is not really parasitic. It's predominantly photosynthetic and symbiotic. And so from an understanding of the evolution of the apicomplexans, this suggests some how that evolution it could be a missing link in our understanding of apicomplexan biology. It also answers a lot of questions that marine biologists have been asking since we've started to mass sequence. So environmental DNA since we've started to sequence it in coral reefs, always one of the first sequences. So those sequences come back and we make kind of OTUs, operational taxonomic units, and we compare them to sequence databases, and they tell us, oh, this is a sequence, there's a sequence of a fish, and a sequence of a cnidarian, and a sequence of this, that, and the other thing, and poriferans. And always one of the most abundant sequence diversity categories in the sequencing of environmental water sources in the marine environment was apicomplexan, which was apiconfusing, because where were all of these parasite genes coming from? And this is kind of the answer to that. It's that they're not parasite genes, they're part of the coral. And as a segue to next week, one of the very cool things about, so that discovery by UBC was a couple of years ago, 2 years ago, I think 2019. Here's a paper from late 2020 where a number of Russian scientists have gathered together and have started naming some of these new lineages. And so I might use that as an entry point next week when we talk about the species naming short assignment that you've got. I might use this as an entry into that. So I'm going to jump back over and take a look at the cat. Is he recording? Yes, he is. He's always recording. This is always going to be a either synchronous or asynchronous. You have that choice in attending. I'm a tiny bit confused in this example. Is that a type of coral or growing out of the coral? Oh, I think you're maybe asking about the loop, the video that was showing. That was a sped up view of polyps emerging from their calcium carbonate homes. That's what that was. Okay, it's 20 past, so I'm cognizant of the fact that you have other things to do. I hope that you have great weekends. I hope that you take care of yourself and do good things for your brain and your body. And we will see you all together again on Monday. And take care of yourself, take care of each other. And I am going to hang around. I'm going to turn off the video, stop the recording, mute my mic for a second, but I will come back in a second for anybody that's interested in, has some more questions or is interested in chatting. So have a great weekend. Take care.